AP 120, Chapter 10, Topics Hearing and Equilibrium. So hearing and equilibrium are localized to the ear. That is where we detect those sensations. And the ear gets broken down into the external ear, the middle ear, and the internal ear. So the outer or external ear, the flap of material that makes the outermost portion of the ear is called the auricle. So this is the auricle on the outside, and then it leads to a tunnel referred to as the external acoustic meatus. So sound waves get funneled by the auricle into the external acoustic meatus, and those sound waves travel down to the end and then collide with a membrane structure called the tympanic membrane. And that causes the tympanic membrane to vibrate. So it's sound waves, these pressure waves in the air that collide with the tympanic membrane, causing the vibrations. From there we go to the middle ear. The middle ear is another air-filled chamber. Within it we have the three auditory ossicles, and they are the small bones that are needed for the hearing sensation. Also found associated with the middle ear is this tube that connects the middle ear with the pharynx. And this is important. This allows us to equalize air pressure on both sides of the tympanic membrane. Normally, this is just minor adjustments that we're not even aware of, but sometimes when you're, say, flying in an airplane or traveling up a mountain really quick, you can actually feel a sort of a popping sensation, and that is the auditory tube opening to allow equalization of air pressure on both sides. And if that didn't happen, that would muffle the vibrations of the tympanic membrane, and then it would be more difficult to hear. All right, the three auditory ossicles are the malleus, incus, and stapes. So when the tympanic membrane vibrates, it causes the malleus to vibrate. Malleus then causes the incus to vibrate. Incus causes the stapes to vibrate. And then the stapes pushes into a membrane structure called the oval window that leads to the inner ear. Now, why do we have these three tiny bones interconnected and vibrating? Well, it turns out they amplify the vibration. So they make the vibration that started with the tympanic membrane more powerful, so letting us to hear softer sounds. Then, of course, the oval window is the connecting point to the inner ear to pass the vibrations into the inner ear. But the inner ear is filled with fluids, so now we are turning what was originally pressure waves in the air to pressure waves in fluid. Here is the malleus, incus, and stapes. They are the smallest bones in the human body. All right, the inner ear or internal ear has a inner membrane structure found inside of an outer bony structure. The inner membrane structure is called the membranous labyrinth. See, it runs all the way through. And the outer bony structure is called the osseous labyrinth. And the inner ear is found inside the temporal bone. Now, there is fluid between the osseous labyrinth and the membranous labyrinth that is called perilymph. Peri, around, so it's around the membrane. And then inside the membranous labyrinth is more fluid called the endolymph. So inside the membranous labyrinth is endolymph. We can break the inner ear into three pieces. We have the cochlea. The cochlea is sort of the snail shell sort of shaped portion. That is where hearing is detected. Then we have the vestibule, sort of this blockish area in the middle. The vestibule is where static equilibrium is detected. And then these three tunnels here are referred to as the semicircular canals. Again, there are three of them, and they help us detect dynamic equilibrium. Semicircular canals detect dynamic equilibrium. All right, so the cochlea. Cochlea starts with the oval window, and the oval window leads to what's called the scala vestibuli. This is the somewhat more superior chamber that is, of course, filled with fluid. So when the oval window vibrates, it sends those vibrations, those pressure waves into the perilymph found in the scala vestibuli and allow it to start traveling up the cochlea. Then we have a lower compartment on the other side called the scala tympani, and it is also filled with perilymph, and it ends in a membranous structure called the round window. And so any leftover sound waves or pressure waves will travel along the scala tympani and then get dissipated through the round window, so we don't get any feedback. Then, of course, is the very important inner membranous membrane, and within it is, of course, fluid, the endolymph. We refer to this when it's in the cochlea as the cochlear ducts. The membranous membrane is the cochlear duct. What separates the scala vestibuli from the inside of the cochlear duct is a membrane structure called the vestibular membrane. And then what separates the cochlear duct from the scala tympani is another membrane, the basilar membrane. And so what happens is those pressure waves in the fluid will push through the vestibular membrane, causing pressure in the fluid of the cochlear duct, and then that would collide with the spiral organ, and the spiral organ is then where we would detect the sensation and interpret it as sound and hearing. There's a close-up of the spiral organ. It includes this cartilaginous structure called the tectoral membrane. It includes these epithelial cells that are providing support, and it also includes the hair cells. The hair cells are the receptors for hearing. Hair cells have tiny little hair-like structures embedded in the tectoral membrane. And so what happens is when those fluid pressure waves arrive, they collide with the tectoral membrane, causing it to vibrate. And that vibration bends the hairs of the hair cells. That is the sensation the hair cells detect. And then they pass that information on to the sensory neurons that then send the signal to the brain. So hair cells are mechanoreceptors. The physical change in the position of the hairs of the hair cells is what caused the sensation. So it all starts with these sound waves arriving in the external acoustic meatus. They collide with the tympanic membrane, causing it to vibrate. Tympanic membrane's vibrations cause the malleus to vibrate, cause the incus to vibrate, cause the stapes to vibrate. Stapes push in through the oval window, leading to the pressure waves in the fluid of the scala vestibuli. That will then travel up the cochlea until it reaches the appropriate area. Then the pressure waves will pass through into the cochlear duct, will then hit the spiral organ, causing the hair cells to bend and generate the signal going to the brain. And then finally, any leftover pressure waves will pass through completely the cochlear duct into the scala tympani and get dissipated through the round window. And that's how hearing works. Turns out the cochlear duct is tuned, as it were, so that higher frequency sounds enter early on, medium frequencies later on, and then low frequencies need to get all the way to the end of the cochlear duct. You can sort of think of it as like a keyboard or a piano where different keys are tuned to different frequencies. Here, the cochlear duct has different regions tuned to different frequencies. All right, so the signal goes through the vestibulocochlear nerve into the brainstem, and then from there to the thalamus, and then to the primary auditory cortex in the temporal lobe. Turns out that what you hear with your right ear would go to the left side of your brain, and what you hear with your left ear would go to the right side of the brain. All right, equilibrium. There's static and dynamic equilibrium. Static equilibrium is detected in the vestibule in structures called the utriole and saccule. So within the vestibule, we have these smaller structures, and that is where static equilibrium is detected. What is it? Well, it is basically the position of the head compared to gravity. And then dynamic equilibrium is detected in the semicircular canals. Dynamic equilibrium is detecting the motion of the head and the body, so the movement of the head and body, which is important for maintaining balance. So static equilibrium within the vestibule, we have the structures called the macula. In the macula, you have epithelial cells supporting hair cells, and then you have a gelatinous structure filled with tiny crystals called the otolithic membrane. And so the hairs of the hair cells are embedded in this gelatinous mass called the otolithic membrane. And when you change position of your head, gravity will pull on the otolithic membrane, causing it to shift position to ooze in the direction of gravity. That will bend the hairs of the hair cells, and that is the signal, the sensation that then gets passed on to the sensory neurons. So what kind of receptor is hair cells? Dooby-dooby-doo. It is mechanoreceptors. Bending of the hair cells is what generates the signal. And this, of course, helps us know up and down and our relative position to gravity and down. All right, dynamic equilibrium, again, is found within the semicircular canals, specifically in the widened area of the canals called the ampullas. And in the ampullas, you find a cupula, where you have epithelial cells providing support for hair cells. You have this cartilaginous structure called the cupula, and then the hairs of the hair cells are embedded in the cupula. And also remember that the semicircular canals are filled with fluid, so the cupula is surrounded by fluid, the endolymph. And then when your head is moved, is turned rapidly in space, up, down, right, left, whatever direction, this will cause a current in the fluid within the semicircular canals. That fluid, as it moves, will hit the cupula, bending the cupula, and that causes a bending of the hairs of the hair cells. And again, it's that bending of the hairs of the hair cells that is the signal we detect. So again, what kind of receptors are hair cells? Dooby-dooby-doo. They are mechanoreceptors. Now, little children love dynamic equilibrium. They love playing with dynamic equilibrium and messing with it. And they do this by spinning in circles really, really fast. That's right. When you do that and you get dizzy, that is your dynamic equilibrium sensation becoming overwhelmed from a signal. And that is it for this part of Chapter 10.